Ladies and gentlemen, welcome to the afternoon session. I hope you enjoyed this morning's session as much as Dr. Feinberg and I did. It's a compliment to the panelists and the moderators that we had such a lively discussion, broad-ranging, and yet even gained some time. And so we're starting 15 minutes earlier with panel 3, which actually be really at the heart of the matter, because now we will actually start discussing in detail the proposed framework. So it's a great pleasure for me to introduce the two co-moderators for this panel. Dr. Amy Patterson, already met her this morning and we all know her very well, the Associate Director of Science Policy here at the National Institute of Health. And we're also very happy that Dr. Samuel Stanley can be here. He had some challenging family circumstances to get here, but we appreciate it even more that you could actually be here because you're, I think, uniquely qualified to lead this panel. Dr. Stanley is not only president of Stony Brook University, but also the chair of the National Science Advisory Board of Biosecurity that we heard about so much already today. So in order to get a full discussion also with the audience in panel 4, as we announced this morning, we again try to speed up a little bit and will, for this particular session, cut short by 20 minutes the discussion both with the panel and the audience. But don't worry, all of this is meant to give you ample time when we actually are in panel 4 discussing the 4 special cases to give you an opportunity to react to these matters. So I would like now to invite Dr. Patterson to begin the panel with a presentation of the proposed framework for guiding U.S. Department of Health and Human Service funding decisions about highly pathogenic avian influenza H5N1 gain-of-function research. That must be the longest title I ever pronounced. Dr. Patterson.
Thank you, Dr. Dijkgraaf. Actually, I'm going to turn to my panel co-moderator, Dr. Stanley, and ask him to say a few remarks to put this a little bit in context.
So thanks so much, Amy, and it's a great pleasure to be here and to join this very distinguished panel. Our purpose today is to present and hear perspectives on a proposed framework for scientific funding agencies in the U.S. Department of Health and Human Services, a framework that they would use to try and evaluate and determine whether research proposals for gain-of-function research involving H5N1 are acceptable for funding. This proposed framework really describes a process for review. It's essentially a conceptual framework, though, at this point in time, designed to guide health and human services and HHS funding agencies in determining on a case-by-case basis whether the risk posed by this line of research is justified. I want to emphasize again that this is a work in progress. You've heard it before, but I want to say it again. And the purpose of presenting it today is to really gather perspectives that will help HHS in really finalizing the framework. They've already heard some reactions already. We presented this at the last NSAB meeting, which I had the opportunity to chair. We had a very vigorous discussion at that point in time. But again, we're very much looking forward to what this audience has to say as well, and particularly what our panel has to say. And they've been selected because they represent very diverse professional disciplines, different countries, and intergovernmental agencies that have a great stake in this research and its results. So the flow of this panel, as you heard, is going to be a little bit different from the previous 2 panels because first Dr. Patterson is going to give a presentation on the proposed framework, and then we'll give the panelists a chance to comment on that proposed framework. And again, they each have questions that they've been asked to address in response to Dr. Patterson's presentation. And of course, we'll conclude by opening the floor to a discussion with the audience. So without further ado, I'd like to invite Dr. Patterson to give her presentation of the proposed framework.
Thank you, Dr. Stanley. Good afternoon, everyone. So I have the pleasure of presenting to you what is truly a draft framework about how HHS might approach its decision-making with regard to research that affords gain-of-function attributes to highly pathogenic avian influenza H5N1. So we heard this morning that one of the central public health questions about this virus is, could the currently circulating forms of the virus evolve to spread efficiently in humans? And we also heard this morning that much of the research that's designed in this arena is seeking to identify the genetic determinants of pathogenicity, transmissibility, host range specificity, and viral resistance to therapeutics. When we talk about the research in this area, gain of function is but one of any number of methods that are used. And gain of function in this context, I just want to emphasize, it's a term that is broadly used to mean giving a new or enhanced activity to a protein. But in this context, we are using it to refer to experiments that generate influenza viruses with enhanced transmissibility, enhanced pathogenicity, and or altered host range, and then testing those viruses in vitro or in vivo. It's also important to note, and I saw one of the editorials in the press recently about this, the author was observing that a loss of function experiment can sometimes, surprisingly, lead to a gain of function. So we are really talking about what is reasonably anticipated in the experiment, but also what might result as an unanticipated outcome. As Dr. Fauci mentioned, and as did the other panelists, the high mortality rate that has been associated, at least within the reported human cases since 2003, heightens our concerns then about doing experiments that would enhance the transmissibility in particular of this virus. And it leads us to ask the very fundamental question: are gain-of-function experiments necessary to address the critical public health questions regarding this virus? So the issues that the Department of Health and Human Services has been wrestling with and that we want to put on the table to discuss with you today and tomorrow and in the weeks to come: is there any HPAI H5N1 gain-of-function research that should not be funded by HHS? And the flip side of that coin, what are the risks of not doing such research? What are the principles and risk assessment framework that should guide decisions about the types of research, if any, that should be funded? And for such research that is found acceptable for funding, what review and oversight would be appropriate, and what biosafety and biosecurity measures would be appropriate? And a very fundamental question that was posed at the beginning of our session and recurs throughout each panel: should we be pursuing this research if we believe it cannot be openly communicated? And in particular, and very pointedly, for the Department of Health and Human Services, with its mission to advance public health, should HHS support research that we do not have every expectation at the outset would be openly communicated and shared? So those are the issues that we've tried to address and wrestle with as we develop this framework. So I'm going to walk you through the framework. It sets out its own scope and applicability, but importantly, it identifies several criteria that must be met in order for HHS to contemplate funding research in this arena. And it goes further to describe characteristics of a subset of that research that might warrant yet another level of review, a multidisciplinary review that includes the security community and others, or department level review. The framework also importantly underscores the critical need to assess and manage biosafety, biosecurity, including the dual-use risks. Before walking you through the framework, it's worthwhile to pause. Some of our previous speakers have already acknowledged this. Research with high-path H5N1 already is subject to a whole host of regulations and rules in this country and in many countries. In the US, what's listed on this slide give you a few examples of the types of rules and regulations that are already in place. This is a select agent. It's regulated as such. There are particular biosafety considerations that are in place for both the physical and biological containment of this agent. In March of this year, the U.S. government issued a policy for oversight of life sciences, dual-use research of concern. Work with this agent falls under the requirements of that policy. And in addition, we have export controls, including international export control agreements, and has been mentioned in the morning, the Biologic Toxins Weapons Convention. These are just examples of the rules and regulations that try to speak to the concerns of safety and security and dual-use concerns. The current review process for this research, at least in the U.S., is that it is subject to several levels of review, both at the proposal stage and on an ongoing basis. So at the outset, it will undergo peer review for scientific merit. It will undergo funding agency review for the dual-use research of concerns issues, as now required by federal policy. It'll have to be registered and complying with select agent programs, and it will be evaluated for programmatic consistency and priority within the overall funding agency portfolio. In addition, at the local level, the Institutional Biosafety Committee is entrusted with ensuring the appropriate biosafety conditions, and compliance with appropriate biosafety conditions is a term and condition of funding for this research. So now let's move on to the proposed framework. I want you to understand that this proposed framework is built on top of the current existing rules and regulations and review processes. And it aims to ensure an additional review, a second look at these proposals that can be reasonably anticipated to generate HPAI H5N1 viruses with enhanced transmissibility, enhanced pathogenicity, and/or altered host range. And it applies not only to research proposals, but ongoing research projects that are reasonably anticipated to confer these attributes to influenza viruses expressing the virulent form of the hemagglutinin gene from highly pathogenic H5N1. So this is the scope of the framework, the types of experiments to which it would apply. It does not apply to routine characterization studies of naturally occurring HPAI H5N1 viruses that are neither intended nor can be reasonably anticipated to generate gain of function. And by characterization studies in this context, we are including sequencing, testing of antigenicity, antiviral drug susceptibility, transmissibility, and pathogenicity. And in this context, we're using the phrase naturally occurring to refer to mutations that arise in nature or through a natural process, and were not engineered by researchers or obtained by serially passaging a virus. And the rationale for exempting, if you will, the routine characterizations of viruses that are occurring in the wild is to ensure that the properties of these viruses can be determined as efficiently and quickly as possible. This is probably, well, it might be visible to you, legible to you. It's also in your handout, but let me just walk you through. This is a schematic of the basic parts of the framework, and we'll walk through some of the more critical elements of it in just a moment. But we start with the submission of a research proposal to the funding agency up here. It will undergo normal peer review for scientific merit, as well as review for the dual use issues as required by the March policy here in the States. The funding agency will determine if the proposal, if it passes peer review, it's undergone dual use review. The funding agency will then ask the question, does this proposal fall within the scope of the framework, the scope we just walked through together? If it does, the funding agency will ask a second question. Does the research fulfill all 7 criteria to be considered acceptable for HHS funding? And I'm going to walk you through those criteria in just a moment. The funding agency will also consider whether the proposed experiments have some additional characteristics that would warrant a more interdisciplinary, multidisciplinary look at the department level that brings in the security community and others. And we'll walk through those characteristics as well. And I want to emphasize every aspect of this is on the table, and we really want your perspectives. Is this too restrictive? Is it too broad? Does it not hit the right notes? Have we left something out? For that subset of proposals that are referred to the department, so the second step in this process, the department will, of course, look at the funding agency assessments. It's going to consider whether there are any additional factors for risk mitigation that would be important to employ. And it's going to decide whether there's additional expertise, subject matter expertise, that needs to be brought to bear. Ultimately, at the end of this process, whether the decision is made at the funding agency level or for projects referred to the department, there are a couple of outcomes. It could be funded with particular terms and conditions for award. It could be determined not eligible for funding because it doesn't meet the principles and criteria. And as we'll discuss in a moment, it could be considered highly meritorious, but perhaps something that may generate results that could not reasonably be shared, something that more falls into the ilk of the work of other departments not aimed at advancing public health per se and along the traditional lines of HHS research. In that case, we would refer it potentially to one of our sister departments or agencies for their consideration. But we'll talk about each of these in turn. This slide shows you the 7 criteria that we are currently contemplating as using for making decisions about what research in this arena to support. And I'm going to very briefly walk you through each of these criteria. The first criterion states that the research addresses a scientific question of high significance to public health. And at first that sounds, well, that's obvious. Shouldn't all the research that you fund meet this criterion? And the answer to that is, of course, yes. But I think as we heard in the discussions this morning, because of the potential risks posed by this research, or some of this research, there's a sentiment that perhaps the bar should be a little bit higher, that it's important that the fundamental questions to be addressed by the project not only have scientific merit, but a high degree of relevance for advancing public health, and that we be able to articulate that relevance. The second criteria, and I'll say up front, this has been a very controversial criteria, not only when we presented it to the NSABB, but within the U.S. government ourselves, talking amongst one another. The second criteria states, the research does not intend, nor is it reasonably anticipated to yield high-path avian influenza H5N1 experimental virus that has increased transmissibility, pathogenicity, or expanded host range, unless there is evidence that such a virus could emerge through a natural evolutionary process in the foreseeable future. And we've heard a lot of comment about this criteria. People ask, well, what's evidence? What constitutes evidence? What constitutes the foreseeable future? Foreseeable in whose eyes? Some of the concerns that we've heard this morning, and we've heard for many months now about this research, is that it's perceived as creating something that nature would not on her own engineer. So opening a Pandora's box, if you will. Others say that if we can think of it, surely nature can do it. And that if we don't push the envelope of knowledge and understanding the fundamental biologic attributes of these viruses, we put ourselves and the world at a considerable disadvantage. The concept, we may not have the words right here, but the concept that we are trying to capture in this criteria is that the risks with gain-of-function research may be unjustifiable if they advance the understanding of strains that exist or are likely to emerge in nature, and that the risks may not be justifiable for research that generates viruses with pandemic potential that are highly unlikely to emerge in nature in the foreseeable future. I think it's fair to say that there's a world of judgment inherent in almost each of the words used in this criterion. I've also heard that this criterion is unnecessary because the first criteria captures it. The research addresses a scientific question of high significance to public health, and that we should not be limiting ourselves a priori about the tools, the approaches, and the findings that we may discover that could shed light on this. Criterion 3. And again, this is something that we heard quite a bit of discussion about in the previous panels. And this is the notion that there are no feasible alternatives to address the same scientific question in a manner that poses less risk than does the proposed approach. So again, acknowledging that there are inherent risks involved with these experimental approaches that confer new properties on an already infectious agents, this criteria would require that a careful consideration, a thorough exploration should have been given to alternative approaches. Are these available? Would they adequately address, with the same degree of rigor, the same scientific question in a less risky manner? Criterion 4. Biosafety risks to laboratory workers and the public can be sufficiently mitigated and managed. And so funding for these studies would only be awarded to institutions that are compliant with the select agent regulations, use appropriate lab practices, procedures, and equipment, adhere to all the training requirements for personnel, conduct regular biosafety reviews, enroll researchers in appropriate occupational health programs, and have access to the requisite high-quality research facilities and containment barriers. We also put on the table the notion that consideration should be given to whether some of this research may need to be conducted only in designated facilities. Criterion 5. Biosecurity risks can be sufficiently mitigated and managed. Biosecurity risks, including dual-use risks, can never be eliminated entirely. But this gain-of-function of research, according to this criteria, should only be funded if there are appropriate physical, personnel, and other security measures in place. And this includes, again, compliance with the select agent regulations, the physical security measures, the personnel security measures, personnel reliability, and compliance with the dual-use research of concern risk mitigation measures now required by funding agencies. Criteria 6. This is also one that's received a lot of discussion, at least the concept that underpins it, the principle that underpins it. This criteria states that research would only be funded in this arena if the research information that it generates is fully anticipated to be broadly shared in order to realize its potential benefits to global health. A number of our speakers and members of the audience have emphasized that preventing and managing outbreaks of this virus is a global challenge and one that requires international cooperation and sharing of information. It's an absolute requirement. We heard already references to the WHO Pandemic Influenza Preparedness Framework as just one example of a network that simply doesn't work without trust, without collaboration, without sharing. And as a general matter, HHS would only fund research that it reasonably anticipates at the proposal stage is going to generate information products and technologies that can be openly communicated. That's how the benefits of the research will be realized. That said, I think it's fair, and it has been mentioned here today, that it's impossible to anticipate all of the results that will be generated by a research proposal. Unanticipated results may occur, and it may be necessary to require that certain proposals employ additional risk mitigation measures, such as more periodic DIRC reviews, periodic reassessments of the containment measures as a term and condition of funding. But as a general matter, I just want to emphasize that at the outset, when we fund and support this research, it is our every expectation that the research results would be broadly shared. And finally, criterion 7, and I should remark that this criterion is not unique for this area of research. We felt, however, it deserves an explicit articulation. This criterion is also true, for example, for forms of human subjects research, forms of investigational new drug research. This is the concept that the research is supported through a funding mechanism that facilitates the appropriate level of oversight of the conduct and communication of the research. In making its assessment of whether a research proposal meets these criteria or not, the funding agency, as well as the department for that subset of proposals referred to the department, is going to be asking the question, what are the risks inherent with the intrinsic nature of the agent used in the proposal? So the starting viral strain transmissibility and pathogenicity and host range, but also the risks associated with the experimental manipulation that is proposed, the likelihood that the virus will become more transmissible, pathogenic, or have an expanded host range. And at the funding agency review, they will also ask whether the experiment described in the proposal possesses or can be anticipated to generate certain characteristics that, if present, would warrant a referral to the department for review. And these characteristics are shown here. So the framework proposes that high-path H5 research would undergo a department-level review if these studies intend or are reasonably anticipated to increase pathogenicity, virulence, and/or transmissibility of a virus in mammals. So this is mammal-to-mammal transmissibility. Or if it would disrupt the induction of the host's innate immunity, interfere with the effectiveness of a vaccine, confer to the virus resistance to clinically or agriculturally useful prophylactic or therapeutic interventions, or facilitate the virus's ability to evade detection methodologies. Again, I want to emphasize that this part of the framework, too, is up for discussion. Is this too restrictive? Is it too broad, too permissive? Does it impose too much oversight, too many layers of oversight? Everything is on the table for your discussion. So what will the department do when it gets these proposals? They'll review the funding agency assessment. They'll bring to the table, if needed, other expertise, for example, expertise in security, intelligence, preparedness, and response capabilities. They'll determine whether additional measures are needed to mitigate risks. They have the opportunity to take a bird's eye view, take into account the overall HHS portfolio of research in this arena. And they will give a determination back to the funding agency whether the proposal is acceptable for funding. And again, the possible outcomes of this review are that it's acceptable with the usual terms and conditions or with additional terms and conditions, that it is not acceptable for HHS funding because it really falls outside the ilk of the normal type of public health research that we typically fund, in which case it might be referred to another federal agency that would be interested in it, or it is not acceptable for HHS funding because it simply does not comply with the criteria. Much of this morning was devoted to the global health implications of this research, and it really calls for broad input on this proposed framework, as well as the topic in general. And as Dr. Stanley mentioned, at the department's request, the NSABB discussed this framework on November 27th. We are also tasking the NIH Recombinant DNA Advisory Committee, which over the past several decades has provided seminal advice on appropriate biologic and physical containment measures for engineered infectious agents, to provide their recommendations on the appropriate biosafety measures. And the multidisciplinary and multinational perspectives solicited during this meeting and in the comment period to follow will be taken into careful consideration by us as we move forward to try to finalize how the conceptual framework that we will use in approaching these proposals on a case-by-case basis. I did want to say that we are trying to optimize the amount of comment and question period during this meeting. Perhaps not all of you will have the opportunity to come to the mic, or when you get home you'll think of a brilliant question that you didn't ask during the meeting. So we are going to have a comment period. Several of you have come up to me during the course of the meeting wondering if a little bit more time could be allowed. And so we are extending it through January 10th. And we will be posting on one of the slides later the mailbox if you want to submit comments directly on the framework. We would like to provide some guideposts and illuminate a path forward about this research one way or the other. And I think there's a certain sense of urgency in the field. That said, however, we really do want to hear your comments, and we look forward to them. The pencil shown here, not just as mere decoration, but I mean it quite literally. This is still written in pencil. And we want your perspectives on the framework. Specifically, we hope that you'll address, are there any risks that are not covered or spoken to in the framework, including those identified during panel 2? Is the scope of the framework appropriate? Does it capture the right types of experiments? And importantly, are the criteria, those performance measures that really are an articulation of a set of principles, are they the right ones for guiding our decisions about this field of research? And of course, any other comments you'd care to make. Sam?
So Thank you, Amy. So I'd now like to turn it over to our panelists to hear their comments about the questions for discussion. I'll remind them again, 5 minutes. You'll have a chance to weigh in hopefully later on as well. And we'll begin with Daniel Altman, the Head of Pathogens, Immunology and Population Health at the Wellcome Trust in the UK. Dan.
Thank you. Hello. So for those of you who aren't familiar with the Wellcome Trust, we're quite a large European funder of biomedical research and fund quite a lot of influenza research in the UK, Africa and Southeast Asia, including quite a lot of H5N1 programs. None of them are gain-of-function programs, not because we don't support it, but because nobody, to the best of my knowledge, has come forward with a proposal of that type. So we have no specific updated policy of our own in this area. So in view of the kind of rather tough chairing this morning, I thought I'd better read the instructions and do what was asked. So which was to go away and read this draft carefully and comment on it. And it felt invidious to be asked to come here as your guest in the festive season and be rude about your homework assignment. But I'll try and do it not in a spirit of being smug or irritating, but in a kind of heartfelt way, because we haven't done this policy draft at Wellcome, but we do many similar ones. And the way that we would go about it at Wellcome is that the scientists would look at it and draft something, and the medics, and the policy team and the legal team, and we'd spend many months on it and present it to the Board of Governors and approve it and go out and have a big dinner and feel very smug about it, and then put out the call for proposals and then get the proposal coming in. And the first proposal that came in would blow an enormous hole in everything we'd ever written because it wouldn't match our criteria at all and we wouldn't know what to do with it. So it's in that spirit that I hope you won't mind me looking at the document and seeing how we would deal with something like this. And I think the first thing that I tried to do was read it as I would in the Wellcome Trust building, where I've occupied a number of different roles, both as a funding applicant, trying to deal with the instructions and grapple with them and do the right thing, as a grant panelist, trying to apply the instructions and work out what to fund and what not to fund, and now in the last few years, as a grant funder, trying to decide how to spend Henry Wellcome's money wisely and do the right thing. And so when I looked at the criteria, the first thing that interested me was the scope of the document and what's in and what's out and how that was decided upon. Because it says clearly that clearly it has to be limited by some kind of ring fencing, and that experimental gain-of-function mutants are in, but naturally occurring mutants are out. And I was wondering again how I would deal with that as a grant panelist, because what would happen if one of our programs in Vietnam had started doing complex laboratory experiments with a naturally arising isolate that proved to be transmissible between mammals or between mammals and non-human primates? Why would we not want to discuss it within the framework of guidelines like this, and why would we seek to exclude it? So that was of interest to me. I agreed with the part about, we've discussed about needing to discuss the risks of not doing the research. I think we can't be lily-livered about this. We either do it or we don't. And what we can't do is pretend that we can do it by doing modeling experiments or genomics experiments. It needs to be done properly. I agreed with the discussions about the need to educate the public, because I think as scientists we're very vulnerable to those accusations of doing doomsday research. If we don't take the time and the trouble to explain carefully to the public what we're doing and why we're doing it. And at Wellcome Trust in recent years, the funding for biomedical research has remained rather flat, while our funding for public education has gone up dramatically, he said only slightly bitterly. So as I read the document, I think there are a number of things there which would be very controversial, because I think we're trained to look at these things as scientists and not as lawyers. And yet if one was sitting on a panel trying to evaluate phrases like foreseeable future, sufficiently mitigated, no alternative approaches, I wouldn't know where to start as a panelist trying to assess those things, who decides and on what basis. So for my taste, there would be room for further clarification. perhaps the biggest one that I'd look at would be the part about the virus's ability to evade detection methodologies. It's a kind of catch-22 of lacking a priori knowledge and not knowing what to look at if we haven't looked at it. We barely know anything about innate immunity to influenza virus. How are we going to know if it's not there if we don't study it? So to finish, I think we need something that's future-proof, that facilitates the research rather than impedes it, and we need to communicate that cause to the public. Thank you.
Thank you. Next, we're going to hear from Marianne Dunker, who's Director of Public Health, Ministry of Health in the Netherlands.
Thank you. And thank you very much for this invitation to share in this meeting. I find this proposed framework very significant and a very courageous first step of the United States in this arena, and very necessary to start a discussion. And as Dr. Fauci said this morning, the process is at least as important as the concrete decision rules. And I'll kind of look into the process also in my comments, because I find it also very difficult to judge this framework, because as Amy Patterson also pointed out, it is part of the particular set of rules and instruments that are present in the United States. And I find it very difficult to judge the exact fit of this part of the whole structure in the whole of your policies. So I'll refrain from very specific comments, but I have some remarks. And when I look back at a year ago, and I recall the first discussions on the publication of Fouché and Kawaoka and their colleagues, I remember a kind of shock reaction in the public health research community. Many were surprised and even indignant, and I seem to have found a little bit of that even in this morning's discussion. This surprise and this indignance that others are interfering with their studies and their publications. However, in other domains, this is not entirely new. For instance, we license research on genetically modified organisms, we review stem cell research, we critically follow research on new fertility techniques, and especially in the area of defense research, for instance on chemical or nuclear weapons, we find it perfectly normal that we very critically review research and the publication of that. And I totally agree that in the domain of dangerous viruses, we need similar discussions and similar regulations. And therefore, the Dutch government is in the process of developing a biosecurity regime that, well, it's not similar to your regime, but it has some kind of similar elements. And we are developing a legislation program that ties up and complements the existing laws and regulations in a more comprehensive framework. We propose to develop a data bank that identifies all the locations where highly dangerous pathogens are used. We will also train professionals, biosecurity professionals, not biosafety professionals, at the research institutes and laboratories whose assignment will be specifically to deal with the biosecurity aspects, not just the biosafety aspects. We are instituting a small expert agency with experts that have the assignment to support both the scientific researchers and the policymakers. And we also developed a biosecurity toolkit that helps research organizations and laboratories to assess their own preparedness for biosecurity. And in addition, of course, Robert Dijkgraaf also mentioned that our scientists are reviewing their code of conduct and making that even better fit for the challenges that we face. Well, the first remark that I would like to make is that the Dutch biosecurity regime that we are developing at the moment, which resembles much that happens in the United States, will apply to all potential dual-use research of concern in the life sciences in the Netherlands, not just H5N1 and not just government-funded. And that's my first remark on the framework that we are discussing today. It only applies to H5N1 studies and only to government funding. But this kind of research may be funded in many different ways. We have had some examples. And we wish to ensure that we deal responsibly with all the research that takes place in our boundaries, in our country and in other countries. And also we want to deal with research on other health topics that pose safety and security threats. To us, it is important that we develop awareness, expertise, and oversight measures that apply to all dual-use research of concern in the life sciences. And my question is, I don't have the answer, why are we so particularly concerned about H5N1? Because there are many viruses, many infectious diseases that have the potential as well for misuse and as well for threatening our populations in a very serious way. In view of this broad perspective, I wish to make a second remark that does not apply directly to the framework, but to the discussion that we had this morning. And that is that we are very aware that we cannot and do not wish to position a policeman in every laboratory. And we do not wish to and cannot oversee the work of every scientist. And we do not wish to develop a system that only relies on control and censorship. And from this perspective, we find the role of the scientific community itself of paramount importance. It is absolutely necessary that scientists themselves take responsibility not just for their studies, not just for their publications, but also for the possible consequences of their research. In addition to their right and their duty to enrich the world with their knowledge, we expect scientists to seriously consider the possible threats of their research and to, in case of doubt, initiate multidisciplinary discussion on their own initiative. And only then will we be able to avoid measures that are one-sidedly based on control and censorship. And I stressed on their own initiative, and I stressed multidisciplinary, because I seem to have noticed that public health researchers sometimes have a little bit naive view of the world out there. There really are bad guys out there. There really are rogue states. There really are failed states. There really are laboratories that are not up to standard on biosafety and biosecurity. Read your newspaper. So I really would like to stress very urgently to the scientific community to seriously consider this kind of threats and to seriously enter into the discussion, not just with your own community, your own scientific community, but also with others who can help assess these threats and find a balance in judging them. Now, I come to my second remark on the framework itself, and then I'll finish. And that is what Amy Patterson finished her presentation with, and that's the international aspect of this dual-use research of concern. This research is often conducted in collaboration between scientists from different countries. So on this research, the laws of those other countries will also apply. And apart from the fact that the research itself has implications for the whole population of the whole world, this collaborative aspect of the research project that we are dealing with, I think would necessitate to also in this framework itself incorporate some kind of mechanism to deal with the international collaborative aspect of this kind of research project.
So Marion, is that a good place to stop?
It is. I wanted to summarize, but
okay,
this is
good.
it.
Good, thank you. So next we're going to hear from Keith Hamilton from the Scientific and Technical Department of the World Organization for Animal Health, OIE.
Okay, thank you very much for the invitation to be here today, and quite rightly, because we're at NIH, this is a very public health-focused discussion. But since I'm from the animal health sector, it's probably wise for me to give you a little bit of background from our perspective. Now, I work for an organisation called the World Organization for Animal Health, or OIE for short. It would take longer than 5 minutes to explain why our acronym doesn't match our name, but that's another story. We're an intergovernmental organisation with 178 member countries with the mandate to improve animal health, veterinary public health and animal welfare worldwide. We work very closely with our sister organisation, the Food and Agricultural Organisation, and we work very closely with WHO on issues at the human-animal interface. And just a reminder that HPAI H5N1 is an animal pathogen, and it's called HPAI because it's highly pathogenic in birds. And the way we characterize it as being highly pathogenic is because it has a very characteristic molecular sequence at its cleavage site. So that's been a very important fact in animal health surveillance, because we're not so interested in low pathogenic viruses. We're interested in the high pathogenic viruses, which are usually subtypes H5 and H7. So in addition to the public health concerns, H5N1 is a devastating disease in poultry, leading to mortality rates of up to 100% and massive losses to agriculture and economies. We're very much focused on elimination of the virus in the animal source, and this is the most effective way to reduce the animal health impact, but also has a huge impact on reducing zoonotic threats. So veterinary services stand in the front line of reducing both animal health and public health threats. Now, animal health researchers are focused on working to better understand the determinants of risk for animal health and public health, to develop effective methods for detection and control, and for risk assessment. Gain-of-function research can play a very important role in this area, so it is something that we're interested in. And that the human-animal interface, OIE, promotes research to fill gaps in knowledge, to improve pandemic preparedness, and to inform surveillance strategies in animals. So as Ilaria mentioned this morning, we're able to detect viruses that might pose an animal or public health threat early. But the reality of being able to do this probably lies many years away, but we're working towards that in an incremental process. OIE and FAO got together in 2005 to launch a joint network of expertise on animal influenza called OFFLU. And this network was really established to improve sharing of data between scientists and between the Animal Health Network and WHO, and to encourage scientists to deposit their data into the public domain in a timely manner, so everyone was aware of potential risk factors and disinformation. Recently, the OFFLU network, thanks to David Swain, who's on the panel, has developed a research agenda to highlight priorities for funding influenza research in the animal health sector. Now, this research agenda highlights the importance of studies to better understand interspecies transmission, including between birds and mammals, between mammals, and between animals and humans. Now, from an animal health perspective, we strongly believe that regulation of research on the public health side shouldn't create any unnecessary barriers to legitimate and beneficial research in the animal health sector. And specifically, the HHS Framework is concerned with public health gain-of-function research, and it states that such research should only be approved if it offers benefits to public health. But because animal health researchers also have a legitimate reason to conduct such research for the benefit of both public or animal health, the potential impact on animal health researchers should also be considered. There may be a risk that the HHS framework and criteria may spill over and be used by other bodies funding animal health research, in which case, if we use the HHS criteria, it may be difficult to justify some gain-of-function research that is more focused on animal health. A more general gain-of-function research approval framework would need to account for the animal health research sector as well, and it might be worth having a case study to assess this and assess the impact of such a framework. Now, as a standard-setting body and acknowledging the potential risks of manipulating pathogens, OIE promotes compliance with existing international standards and regulations as a way to reducing these risks, whilst avoiding disproportionately risk-averse reactions. So it's important that any regulation, any research regulation, does not hamper scientific innovation. And last week at the Biologic and Toxic Weapons Convention, our Director General spoke to the Biologic and Toxic Weapons Convention, and just quoting his words, it's a very general presentation, of course. His words are that scientific research is absolutely necessary to develop effective strategies to maintain the fight against infectious disease, To create unnecessary regulation or barriers to scientific innovation would be very dangerous. For this reason, OIE strongly supports Article 10 of the Biologic and Toxic Weapons Convention, which promotes a peaceful use of science. Thank you.
All right. Thank you very much, Dr. Hamilton. Our next speaker is Dr. Yoshi Kawaoka from the Department of Pathobiological Sciences, School of Veterinary Medicine, University of Wisconsin-Madison in the U.S.
Thank you. My comment is not philosophical, rather very specific. So in principle, this framework should serve as a useful guide to identify proposals that will require appropriate administrative oversight. After reviewing the framework, this is my perspective. First, my overall response is that the framework is too inclusive to be functional. This framework will require many proposals addressing the pathogenicity of H5N1 viruses to be sent for departmental review. For example, to elucidate a mechanism by which H5N1 viruses from birds become more pathogenic in mammalian hosts, we test the effect of amino acid changes on pathogenicity. According to the increased pathogenicity criterion number 2, all such studies need to be evaluated at the departmental level. Now, in most experiments, we would introduce mutations into H5N1 viruses that are not pathogenic to mammalian species, and then test the pathogenicity of those mutant viruses. Now, some mutant viruses may be more pathogenic to mammalian host than the parent virus. However, it is highly unlikely that the mutant viruses generated would be more pathogenic than any H5N1 virus that already exists in nature. The focus for the departmental review should be studies that may lead to the generation of harmful viruses that do not currently exist in nature. Now, this straightforward criterion is applied by the Institutional Biosafety Committee at the University of Wisconsin at Madison to dual-use research concern reviews. Mammalian transmissible H5 viruses are good examples of this category of experiment. Now, there are specific issues with the criteria that I'd like to address. The criteria number 1 and number 2 are very judgment-based. What is meant by significant? I think all the research that NIH funds should have significance for public health, criterion number 1. This is a broad conceptual foundation on which all funding decisions are based. I have heard some suggest a near-term application would make the work significant. But those of us who do basic research realize that our progress is incremental and builds on the results of others. Now, applications are derived from the body of knowledge that basic research accumulates. The criterion number 2, unless there is evidence. What constitutes evidence? More importantly, if the HHS framework limits research to experimental outcomes we can predict, we will severely limit the ability of researchers to make discoveries and understand these important pathogens. For example, in 1989, late Rudy Roth found a naturally occurring highly pathogenic avian influenza virus that had integrated a portion of host RNA into its hemagglutinin gene. he found that this virus became lethal to chickens due to this insertion of the host genome. No one would have been able to predict such an event, but findings like this contribute extremely to scientific progress. Criterion number 2, foreseeable future. What does this mean? This phrase is subjective and will make it difficult to arrive at a consensus opinion. Now, finally, I have concerns regarding departmental level HHS review. This is very vague, and there are no specific criteria to explain how the department will make decisions. So in conclusion, I would advise eliminating criterion number 2 or simplifying it to mean only those studies that may lead to the generation of H5N1 viruses that do not exist in nature. More details regarding the departmental review should also be provided. Thank you.
Thank you very much, Dr. Kawoka. I'd like to move on now to our next panelist, Dr. Zarifa Reed, Medical Director of Synanex Therapeutics in Malaysia. Dr. Reed.
Thank you very much. It appears that there are two driving fears behind what we're doing today, and I guess both, if taken just schematically and extremely, appear to drive us in two opposite directions. So one is the fear that gain-of-function research is critical, and fears that restriction, over-regulation, over-oversight will stop critical science, which we all seem to have some general agreement that is needed. Then on the other hand is the fear that gain-of-function research is critical, but what about the possibility, small as it might be, that it could be misused, one, or two, suffer from the unintended consequences or accidents that appear to happen, of which we don't really have a real track of, I guess, was one of the concerns today. And the framework is proposed as a process to try to aid HHS and NIAID in thinking through these proposals in a way that tries to address both these fears, which mentioned earlier is very complex. So I guess my perspective on the framework is try to consider a couple of alternatives, or what I see as perhaps complementary to the framework as well, because I agree with sort of the issues that have been highlighted by the other panelists. And perhaps this is already in discussion in different forums, but maybe the time has come also for the scientific community to consider the development of a sort of a general scientific code of conduct. I hear that it is being developed, it has been developed, it is being utilized in some countries, but I'm thinking of just another model within the clinical research community, such as the Declaration of Helsinki, which was formed by the World Medical Association. In a way, I suppose the spirit of actually the community, which is highlighted by previous panelists, of demonstrating concretely some sort of spirit of self-regulation, self-regulate yourselves or you'll be regulated in a way that you don't like. So demonstrating a kind of commitment to common principles which are important for a community despite our diversity, claims as a common objective this advancement of science for the benefit of public health. So in addition, to sort of complement this declaration of ethical responsibility and conduct for the clinical research community of do no harm, it would be great to think if we could all self-regulate to that degree, and then everything would be great. But on the other hand, the other element of sort of clinical research that has come to evolve is also sort of a set of principles or best practices. And those of you who conduct clinical research probably know this, called the good clinical practices, the GCP principles. And just the point that I wanted to highlight with these principles are that they were actually created and formulated by the actors of the industry themselves, the pharmaceutical industry, that sort of recognize the need to be able to conduct their activities in a consistent, reproducible, and regular manner in a way that they could be regulated. So it was a form of self-regulation, really. And someone brought up in an earlier panel the issue of an incentive for self-regulation. So clearly the industry has an economic, in addition to the ethical incentive to self-regulate themselves. So clearly all of these began as examples of self-regulation, and they were all historically did not arise simply from goodwill, I guess. It was events that drove the Declaration of Helsinki, the abuses of medical research that went on during World War II. Other examples of terrible ethical abuses also drove these instruments of self-regulation. So within the context of the two frameworks, institutions have translated these instruments into ethics, scientific review committees, trial monitoring assessment instruments, data and safety monitoring boards, to ensure that not only is there self-regulation, but there's also this other aspect of kind of monitoring and evaluation that goes on. So I guess this brings up another point with the framework, and this is kind of my final point, is that where should this framework be is another consideration. Should it really be perhaps with some of the institutions? Because clearly the investigators are not sort of freelance scientists. They're attached to institutions that they themselves actually have these instruments. And I can just think of a couple of examples where principal investigators that were conducting these type of research actually took it upon themselves to actually conduct a peer review assessment of the risks of the kind of research that they were doing within their own institutions, and then presented the results of that in a transparent forum, but at the institutional level. So I don't know if that is something that you would want to consider, whether, to actually ask the institutions themselves and put that responsibility, like the responsibility of institutional ethics committees and stuff like that, instead of taking on something like this, which seems quite complex, as was highlighted earlier. Thank you.
Thank you, Dr. Reed. And I'd like to now turn to the next panelist, Dr. Michael Selgelid. Dr. Selgelid is Senior Lecturer and Deputy Director for the Center for Human Bioethics at Monash University, Australia.
So thank you for the invitation, Amy. Some of the points I want to make were already made by other panelists, especially the first two. I start with my perspective on the proposed HHS framework, and that's my perspective as a bioethicist. So I'm trained in engineering, biomedical engineering, but I primarily work as a philosopher concerned with ethical issues associated with biology, healthcare, and medicine. Recent controversial cases of dual-use life science research, such as the H5N1 transmission studies among ferrets that we've been discussing today, and numerous other examples that have emerged during the past decade, indicate that the life sciences are in a situation analogous to that of atomic sciences during the first half of the 20th century, when key discoveries were made that enabled the production and use of the first atomic weapons. This is thus a key moment in history for life science policymaking, and the HHS proposed framework for guiding funding decisions about HPAI H5N1 gain-of-function research is a timely, important example of the kind of new policies that are needed. I thus applaud HHS for this initiative. Bioethicists' discussion of ethical, legal, and social implications of genetics, which has comprised a virtual industry within the discipline of bioethics, has largely neglected potential biological weapons implications of advances in genetics. This is unfortunate because the malevolent use of genetic science and technology may turn out to be the most serious ethical consequence of the biotechnology revolution. The proposed HHS framework is apt because basing funding decisions on analysis of dual-use dangers associated with proposed research should be an integral part of the governance of dual-use life science research. Basing funding decisions on analysis of dual-use dangers should play a role in a broader web of prevention involving other or additional governance measures, including ethics education of scientists, review of research for dual-use dangers, review of studies prior to publication, restrictions on access to dual-use materials and technologies, and legislation, international legislation, prohibiting production and use of biological weapons. Funding bodies should aim to support research most likely to promote the greater good of humankind. It is thus highly appropriate that they take dual-use dangers into consideration when making funding decisions. Suppose that two proposed studies are assessed to be equal with regard to the magnitude and likelihood of benefits, where one of the studies poses significant dual-use dangers while the other does not, or where one of the two studies poses greater dual-use dangers than the other. Other things being equal, it would be highly appropriate for a funding body to prioritize the less risky proposed study over the more risky proposed study. If it is correct to assume that funding bodies should primarily aim to promote net societal benefits, then such a conclusion is rationally required. In cases of studies where risks are especially great and likely to outweigh benefits, furthermore, it would be appropriate for funding bodies to refuse funding altogether, regardless of what the competition is in the way of other proposals being considered for funding. And it should be noted that refraining from funding a particularly dangerous proposal involving dual-use research would be a less draconian governance mechanism than some of the other possible governance mechanisms, such as censorship. Okay, moving on to risks not addressed by the framework. First are risks of studies aimed at characterization of naturally occurring viruses. The framework's scope is limited to studies that intend and/or are likely to generate HPAI H5N1 viruses with gain-of-function attributes. According to the white paper draft framework, studies aimed at characterization of naturally occurring viruses, quote, do not intend nor are they reasonably anticipated to generate novel viruses with gain-of-function attributes. The characterization of naturally occurring viruses does not introduce the risks associated with generating or engineering new viruses, end of quote. That might all be correct, but characterization studies might nonetheless sometimes generate information that will enable others to generate or engineer dangerous new viruses. The point here is simply that characterization studies, like lots of other studies, may pose dual-use dangers that should be taken into consideration by funding bodies, and a framework for doing so is wanted. Two additional risks not addressed by the studies, by the framework, involve research that poses the kinds of risks that the framework is addressed at dealing with, but in the case of other flu viruses that aren't necessarily H5N1 and other pathogens that aren't necessarily flu viruses at all. Thank you.
Thank you, Dr. Selgolid. And our last panelist, Dr. David Swain. Dr. Swain is from the Southeast, Poultry Research Laboratory, Agricultural Research Service with the U.S. Department of Agriculture. Dr.
Thank
Swain.
you very much. Simply, I'm a chicken doctor. So I'll make my comments simple. Many of the panelists have already made some of the comments that I was going to use. In fact, the Rudy Rod example was one I had on my notes. So, as the Department of Agriculture, we have a good relationship with Health and Human Services, both at the NIH level and the CDC level. And we feel that there's a lot of cross-cutting science that is exchanged between both of our agencies through both the competitive grant program and in our in-house research programs. One of the critical things that has been utilized, some of our labs, both at the university level and our lab in Athens, have provided assistance to both NIH and CDC in evaluating viruses as their potential agricultural threat. And I think in development of the framework, the general framework is good because it focuses on gain of function for public health or human health, I would say. But also at the same time, we need to remember that the problem we're having today with H5N1 is predominantly and foremost an agricultural problem, and the research that comes out of both agricultural and veterinary medical labs as well as in human health labs has a profound impact on global health and that controlling and potentially eradicating the virus in countries in Asia and Africa will have a profound impact on the global health, not just for those particular countries. One thing I want to bring up in reading through the framework is for us to be very cautious in the nuts and bolts, because the devil is in the detail. I think Dr. Kuoka very apropos proposed some of his concerns, but also I think I have concerns that we be very specific in the language that we use. And as I read through the documents and heard speakers today, occasionally I heard the term gain of function for public health, but most of the time I just heard gain of function and host species, and that could be misinterpreted very easily that the same would apply to studies that were, for example, looking at host species changes in the avian world, in which we have a wide variety of viruses that may have some of those functions already in the field, and if we generated viruses in the laboratory, would those qualify as gain of function or gain of loss and fall into the same sort of logistics. I think that as HHS moves forward, I would recommend that if they do have proposals that do use farm animals, and part of the assessments that may be gain of functions, they would consult with the Department of Agriculture to make sure there are not concerns that we would have or in the animal health realm would have, that those studies would need additional review by us. I think also I would like to emphasize the role of the institutional biosafety committees and the great role they provide, being trained even for us in agriculture. They're trained through the NIH program in how to assess risk, and that should really be the front line in assessments that we do on the risk of research and what's allowed within context of programs, within grants, as well as in broader programs. Thank you.
Thank you. I couldn't write quickly enough. These were all wonderful, wonderful suggestions, and I just wanted to offer one or two words to clarify a couple of points, because I think it might help in the subsequent discussion. One of the things that was raised by the panel members, some of the panel members, is that this framework, this policy as currently configured, it does not address the broader range of infectious agents, the broader range of dual-use concerns. And I just want to be crystal clear that it is not intended to address the full range of dual-use issues, the full range of agents. We have the first step toward that, the March 29th policy here in the U.S., which encompasses work with 15 agents. High-path avian influenza is one of them, and we will be coming out in the not-too-distant future with a draft policy that speaks to both federal responsibilities, but importantly emphasizes the roles and responsibilities at the institutional level and the important role, the personal role that the individual investigator plays. And this is a much broader policy, and its draft, it will be open for public comment, and it asks a very explicit question about its scope. The other thing that I wanted to mention too is that for those studies that are natural characterization studies that are exempted from this additional funding agency and department level review, as at least currently construed, those proposals still undergo a dual use review. So all of the questions about, and I think it was an excellent point that one of our panelists mentioned, it is true that those studies could offer insights into the mutations. And so the informational risks coming out of that work need to be examined, and those are examined under the dual use frameworks. I just wanted to clarify that this is focused for a particular reason on this particular agent. There's some other tools in the toolbox that apply more broadly, and the U.S. is still in the process of scoping and developing its dual-use policy. And so this conversation has been very helpful. Thank you. Sam?
So now's the time for moderated panel discussion. And let me start. We really do have an international panel, and I want to go again with this concept of how does this framework potentially impact international research, and you mentioned already, Marianne, for example, what you're doing in the Netherlands, but I'd again be interested in the UK and around the world. HHS going out in this area, is this going to drive essentially what's going to happen in other places? And how is this, and what do you think about that?
I think it's a very valuable driver, and all power to it for that. I think I've got no sensitivities about that, and nor should we have. Yeah. Yeah.
I think the same applies to us. But I think to us it's a kind of an example of how you can try to be very thorough and very meticulous, and also very transparent in how you weigh in a very difficult. Make a very difficult decision on a complex issue. We won't take over all of it, I guess, because our policies are just kind of different. It doesn't fit in our structures, for instance. And just in the case, like the research of Ron Fouché, which was an excellent example of research funded by you, I think, but also taking place in the Netherlands, and also our own rules and regulations and laws are applicable. I have not discussed that with my colleagues in the Netherlands, but I would very much prefer, and I think the researchers would prefer that also, if we try to debate the consequences of international research up front instead of at the time of publication. If we find some kind of mechanism to make the context and the regulations very secure for the researchers instead of having to wait what every different country is going to do in the end.
Anyone else have a comment on this at all? No? So let me ask one more question, and then Amy, please go ahead. So Yoshi, let me ask you a pretty direct question. How much of your portfolio of the research you're doing now would be impacted essentially by this framework?
This is one of the many studies that we do. We work on antiviral studies, vaccine development, human immune responses. So we do many things. This is one of those important studies.
My impression from your comments was a feeling, a concern that this would be too restrictive, though, in the area of interest. Is that a fair characterization of your comments?
No, what I meant was in my presentation is that we don't want to send many applications, proposals to HHS so that the proposals are not reviewed in timely fashion. And that's what we scientists are afraid of.
So Yoshi, can I ask a very pointed question? What proposals, if any, do you think should have that second level of review? And could you put to David's point, could you be very precise with the terminology and help us out here?
So as I described, the mammalian transmissible H5 viruses, that's the one, because at least at the moment, we don't have H5N1 viruses naturally circulating, viruses that are transmissible among mammalian hosts. So the experiment that may lead to the generation of such viruses should be reviewed at the HHS.
I was just going to make a comment about the logistics of doing research and in high biocontainment labs, which is a very expensive space. You want to be able to, in a timely way, have things reviewed so you can progress with the research. And I think in this situation, one of the points I was trying to make is that we need to be very careful about what we define as the gain of function and make it very precise so that we don't get regulatory drift. And what I mean by is we start off having a language of very restrictive of transmissible to mammal transmission, but then we broaden that because, all H5 viruses may have infected a mammal. Let's say, for example, you get a virus from a tiger or a cat or a dog from Southeast Asia. Would those fall in that category? And I think that's where we have to be careful, because if you quickly broaden the definition, then you're going to very quickly overload the evaluation system that's going to be at the department level. And I think that's where you need to have empowerment, of course, to the local institutional biosafety committee to be able to evaluate based on a very strict definition what needs to go forward, what doesn't go forward. Because once you send it forward, and if you have too many things coming forward, it's going to bog down the review process. And then if you have multiple institutions who have high containment labs who are scheduling experiments, that becomes a very expensive space to have to cancel because there hasn't been an approval for a particular protocol.
So, Yoshi, can I ask, well, David and Yoshi, when you say mammalian transmissible, can I push you to even be more explicit? Are you talking about sustained mammal-to-mammal transmission? Are you talking about transmission through via respiratory droplets? Or are you just talking about it's been demonstrated to be transmissible?
I'm not sure what you mean by sustained transmission in mammalian hosts. We don't have the system. We only have the system to test the transmission of viruses via respiratory droplets. That's the system.
So, for preciseness of terms, transmissible in mammals by respiratory droplets. That would be the criteria in your mind.
Yeah.
Good. I think I'd like to hear from the audience. So why don't we move to that stage and take questions from the audience about the framework. Are there any? Good. And I'll remind everybody to identify yourself and try and stay within about 90 seconds for your question or comment, and I'll begin with you.
Yeah. I'm Mark Frankel. I'm on the staff at AAAS, where I do science policy. I have some very practical questions. They're not theoretical, they're not philosophical, and perhaps they should be delivered to Amy, that maybe she can help me understand better some of the provisions that have not been discussed yet. But a lot of them have to do with things like clarity and transparency and fairness. One of them has to do with this notion of adding additional risk mitigation measures the funding agency may want to do, or the departmental review, et cetera. And one part, at one point it uses the term that they will do this because they are to designate what these are. In another place it says may recommend. That's could be a significant difference in terminology, so I'd like to get that clarified. With regard to agency and departmental reviews, it says proposals determined to be unacceptable for HHS funding are not eligible for agency support. My question is, will those decisions and reasons for them be made public so that we can learn about the reasoning that went on behind those decisions? With regard to box 2, in making those decisions regarding those criteria, will each HHS agency have its own approach, or is it intended to be a process that's uniform across the department? A couple of questions that raise issues of fairness. It says that the funding agency will incorporate any additional measures into the terms and conditions of the award as necessary. Who's going to pay for those additional measures? Then it says if either an agency or department review results in no funding due to the risk assessment, my question is what route of appeal will be available to a PI or her institution? And then finally, a more generic question. We know that researchers put a lot of effort into these proposals, and the framework is relatively or virtually silent on their role during the course of this process. There's a lot in there about what HHS is going to do, but very little about what's incumbent upon the investigator and the institution. And I'm just wondering, what do you expect from the investigator and the institution during this process while it's ongoing, and why not make it more explicit?
Great. Amy, do you want to just address the one issue around the investigator, what you might envision with the investigator-agency interaction?
Okay, and Mark, I can follow up with you on the others at another time. All good questions. In terms of the investigator role, first of all, as all investigators in this room fully understand, they have a tremendous role in crafting the research proposals. There's often a great amount of interaction, particularly in this research portfolio and others like it, between the program staff and the investigator. So we envision a fair amount of dialogue between the institution and the investigator and the funding agency to understand the experiment, to understand what other modalities have been explored, what the anticipated risk mitigation framework is. So we really look at this as a collaborative process with the institution and with the investigator. And at the end of the day, however, we do have to make a decision about what we're willing to fund or not, and that is ultimately a decision that will rest with the funding agency. but I think we have a reasonable track record of working very well with the investigator community and institutions to try to understand their thinking and to come up with a reasonable path forward.
Ron?
Yes, thank you. Is this on? Yeah. So all of the discussions so far have really evolved around HHS as sponsor of the research. And it's been said that if HHS does not want to sponsor the research, it may still be sponsored in a classified environment sponsored by the Department of Defense or Homeland Security or Energy. Although this framework deals with classifying communication of the data, it does not eliminate nor reduce any of the more important fears around this type of research. The fear that these viruses might emerge in nature, the fear of insiders to do harm by intentional release, or the fear of accidental release from the labs that do the research. I think that restricting this or classifying the research will only make these problems bigger, in fact. All of the transparency and oversight will be gone. Nobody will know what research is sponsored in the US, in the UK, in China, Russia, Iran, or the Netherlands. There will be proliferation of this research, as each country will have its own secret research programs without communicating about them whatsoever. And so when it comes to the benefits of this research, there may be no benefit at all of doing this research in a classified environment. So my question to the panelists, what they feel about classifying H5N1 research if HHS does not fund this research.
Good question. I'm just going to go around. Danny, your comment?
I think I do accept that there will be small subsets of research that are done under classified conditions for good reasons, and I can live with that.
Marianne?
I don't have any opinion on that on this moment.
Okay. Keith?
Yeah, probably a more general comment, actually. We saw this morning the sort of impact of pandemics over the last century and a decreasing mortality rate, and the comment was brought up about strengthening health systems, and probably the best way to eliminate risks and to combat biothreats is to have stronger health services on the public health side and on the animal health side. And research plays a very important role in strengthening those health services, and it has done over the last century. So I think we must promote, as Ron said, some of the benefits of research.
Yoshi?
We are all concerned about pandemic with H5N1, and so considering that, I don't see any experiments has to be classified with the H5N1 virus.
Okay. Zarifa?
Yeah, I have to agree with Yoshi. I think you've just raised a good point, sort of on the other side, another fear, which I don't think we've really addressed, which is to sort of drive the research underground. So maybe it's, it's better the devil that you know than the one that you don't if you classify such research. So.
Yeah, I can imagine numerous cases of research that I would think should be conducted and shouldn't be made public. I don't see why we should think that classified research can't have benefits. Lots of classified research presumably does have benefits. And I don't think we need to conclude that if research is classified, then there won't be any oversight. We should hope that, and hopefully we can trust that the powers that be will make sure that any classified research that's justified is done with especially great oversight.
David?
Yeah, I think I've struggled with the word classified as was brought up earlier. Is that part of the mission or not of the mission? And I think for us in agriculture, we do research for benefit of humanity, and we don't have a mechanism really of doing classified research. So for us, it would be probably a no. That's my personal opinion. And if we did have classified research, would that violate a bioweapons convention? If it's researched for gain of function and transmissibility humans to human, would that be a violation?
Good. So, Ron Ford from the panel. Yes.
Joe Dudley, SAIC. My question is to the respiratory droplet transmission. Would this categorically exclude gastrointestinal transmission or mucosal contact transmissions? The normal route of influenza transmission in birds is oral fecal. Gastrointestinal infections have been documented in human beings. This is my concern.
Yoshi, do you want to comment?
I'm not sure what is your question.
Is the language respiratory droplet, which you said, restricted to that this oversight would be applied only to respiratory droplet transmission experimental context? Using the same virus, could you do it in gastrointestinal with the same sequences and thus avoid the respiratory droplet restriction as a way to work with exactly the same genome, but without the same regulatory oversight?
So what I meant is the virus transmitted through air, that's all. How the virus get to the second animal through the mucosal contact or any other mechanism doesn't matter. Whether the virus transmits via air, airborne, that's what I'm talking about.
Thank you. Good. Yes.
Reprising an old role here somewhat involuntarily, but the framework states that any entity that would conduct this type of research would have an institutional biosafety committee. That would be a welcome step forward. When the news of Dr. Fouchier's experiments first became public, I filed a Freedom of Information Act request with OBA to get the list of currently registered institutional biosafety committees, and Erasmus University is not among them. And this is not the first time where research of a particularly risky type involving influenza has taken place, not funded by NIH, or the other U.S. government agencies, not under the oversight of an IBC. The initial experiments to reconstruct 1918 influenza at the poultry lab in Athens were not reviewed by an IBC either, and there are other instances that I can discuss. So when you state in the framework, this is really probably a direct question for Dr. Patterson, that any entity that received a grant to do this type of work would have an IBC. Is what you mean that they would have an institutional biosafety committee that's registered with the National Institutes of Health and subject to the NIH guidelines and anything else that they would be obliged to follow as a result of that? Or is what you're saying that they would be subject to whatever national guidelines that might or might not sort of resemble an institutional biosafety committee in whatever jurisdiction it is where the research actually takes place?
So thank you, Mr. Hammond. That's a good question. And I think the answer to that, and I'll call on colleagues at NIAID, maybe Dennis or Tony want to speak to this, but what we intend by this statement is that if it's supported by HHS, that it would be compliant with the requirements to have a registered IBC, and there may be additional requirements in place in the country where it may be conducted, but we would require that we have records of the registered IBC and its appropriate constitution. Dennis, I don't know whether you want to add anything to that or not in terms of particularly the international agreements that are in place for work with select agents and the equivalency inspections that go on between here and a country where research with a select agent is being supported by the United States government.
I agree with what you say. I have nothing more to add about it. I do have a question of wouldn't this be covered already under recombinant DNA guidelines, since many of the experiments involve recombinant DNA and the requirement for an IBC?
Yes, that is true.
Yes.
Excuse me, Richard Webby from St. Jude Children's Research Hospital, an influenza researcher. I guess my question is really a clarification. It's not entirely clear for me from reading this as to what actually triggers the departmental review. I think I have exactly the same concerns that David and Yoshi do, that this, if you read this conservatively, as I think if it's the study section that's going to trigger that review will do, you're essentially going to be reviewing 75% of all H5N1 studies in the NIH portfolio. Certainly 75% of what we do. And I think that's, it's very critical as to what, again, it's not clear exactly when that review is triggered and when some of these filtering come into play. Again, I think Yoshi's point is perfect. The transmission is an easy one, right? So we're gain of function from mammalian transmission, these viruses don't have it. But when we come to the pathogenicity, I think there's a a misconception that all H5N1 viruses, a ferret gets a sniff at it, they die. That's far from the truth. We have a little bit of we're a WHO collaborating lab as well. We make ferret serum to follow the evolution of these viruses. We did a quick look back through our records in the past 6 years. I think we've put close to 50 H5N1 viruses into ferrets for ferret production. Only 2 killed ferrets. Most of the rest were asymptomatic. So I think particularly this thing of pathogenicity, gain of function for pathogenicity, needs a lot more work. Otherwise, I can see this just swamping the system and certainly leading to a reduced desire to do H5N1 work.
Do you want to comment or no? Okay. All right. I think actually you were next. Go ahead.
I'm essentially going to echo what Richard said, and my concern. I'm Kanta Subara, I'm an influenza researcher at NIH, and I have the same issues with criterion number 2. It's very hard to interpret what is meant by the pathogenicity or the enhanced host range. Transmissibility is an easier one to understand, and I would support Yoshi's simplification of that statement.
Good, thank you. Arturo.
I want to support Joshi's comment about criteria number 2. I think that it is uninterpretable the way that it's written right now, and some parts of it almost feel like you have to prove a negative, which is it's going to be. That alone is probably going to stop a lot of the work if it stays there. I have 2 other comments. I currently, and I say this with some trepidation because I am still at the NSABB and I certainly do not welcome an enormous amount of work, but it seems to me that the only body really that it should be looking at this thing, the only body with any sort of experience and which is in NSABB, so I don't understand why this thing is going to HHS. Will they then reconvene a body like the NSABB to review this? At least the flu community now has experience with the NSABB. And this is a mechanism, I think, that if these things are going to get reviewed, the only body on the land really that is, I think, currently has the expertise to look at this is the NSABB. And the third thing is that I want to point out to you that, this framework is going to add it's going to add oil to the crankcase of a lot of H5N1 research. Because think about it, it's going to make it, somebody just pointed out, 75% of the H5N1 research in the United States may fall into something like this. And I think it's going to drive a lot of people out of the field. And it is precisely the opposite of what we need to be doing if we're going to be confronting an epidemic. So I really think that a lot of more thought ought to be given to this, because we are potentially doing something that it is, in the long term, very harmful to humanity.
Thank you. So, and I think that does, I think we're hearing, and I think that's why it's so important, essentially, to hear from the people doing the research, to understand, does this have the capacity to put a chilling effect, essentially, on research that I think there's been general agreement today is extraordinarily important. So, I think these comments are well put. So I'm going to go, someone who hasn't spoken yet, and then I'll come back to you, Ron. So go ahead.
Thank you. Nancy Cox, CDC. For those of us who sort of work at the intersection between surveillance, risk assessment, and preparedness, I was sort of a little concerned about some of the panel members' comments about characterizing naturally occurring viruses, because if we have to take all of those studies up to a higher level of review, that will inhibit our normal risk assessment of what is going on out there in nature. And that would really be a tragedy. It would delay our antiviral resistance studies to determine whether or not the viruses circulating in nature are resistant to antivirals. It would slow down our candidate vaccine development and a whole host of other things that are essential for preparedness. So I just hope that by saying this, I've sort of raised the awareness a bit.
Good, thank you. Ron.
A quick comment first to the gentleman that discussed the issue of a lack of IBC in Rotterdam. Clearly, Rotterdam University has an institutional biosafety office. It's not called an IBC. We have different laws and regulation in the Netherlands, but it has been ensured by NIH that our institutional biosafety office complies with the US rules and regulations, just for the record. Secondly, we've heard about the fact that all 7 guidelines in the framework are very subjective, from number 1 through number 7. That means that it's absolutely critical who is going to be vetting the research that's coming in front of that committee. If it is Yoshikawa Oka and Ron Fouché and 5 of their colleagues, it's going to look a little bit different than if we're going to get Thomas Inglesby and 6 of his colleagues to be vetting this research. But I'm very interested in what type of people, what sort of background will be in the committee that will be vetting this research, because that's going to be critically important for what the outcomes will be.
Okay. Yes.
Laurie Garrett from the Council on Foreign Relations. Criteria 2, as I'm hearing more and more discussion about it and rereading, I'm finding myself thinking about a National Academy of Sciences debate between Howard Temin and Josh Lederberg in 1988. And the debate centered on what would it take for HIV to be an airborne virus. It was quite heated, and fortunately at that time, we didn't know how to do reverse genetics and all the things that would have made it possible to do a gain of function assay on HIV in order to try and see what it would take to push it to be an airborne transmissible virus. But certainly that was the nature of the argument, and it was left with Temin saying, I've done a calculus in my head, and I think the odds are 1 in, I think he said 800 billion, something like that. And Lederberg said, fine, I'll lose sleep for both of us. I feel like if I took this criteria to foreseeable future and thought back to 1988 and that debate, would foreseeable future be all the way to today? Would Howard Temin and Josh Lederberg had to debate whether or not to have experiments to look for gain of function for HIV that went all the way out to the year 2012? And what is foreseeable future now with synthetic biology and the possibility of transmitting sequences to a 3D printer? What does that mean for 2015, much less 2020, or the equivalent of Lederberg-Temin debate era to that much further into our future now?
Thank you for your comment, and this will be our last comment or question.
All right, so Daniel Perez, University of Maryland. I would like to say that foreseeable future, there is only one kind of people that can do that. That's psychic readers or palm readers. Anyone else is disqualified from doing such work. But I would like to comment on how specific, if there are new regulations, how specific they have to be, and if they are going to be related to mammalian H5N1 transmission studies. I think that none of us have done sustained mammalian transmission studies. By that I mean the ability of a ferret being naturally infected from an airborne virus to another ferret that gets that virus, okay, from that one that was naturally infected. Most of the studies that we've done, and perhaps some other groups have done something different, but they are restricted to infecting ferrets with a large dose of virus and see whether those ferrets are shedding enough virus that it can be airborne. So it's not sustained transmission, it's just one single event that can be reproduced several times in the lab, but still is one single event. So I think that we have to have a very good definition of how we're going to talk about the mammalian transmission studies. On the other hand, for those of us that keep telling us that we should keep it real, that we should be aware that a rogue states or rogue groups, you only have to, you don't need biotechnology to cause harm. All you need to do is to be in one of those countries in which H5N1 is endemic, that do have fundamentalist groups, and they may trace those outbreaks of high mortality. They may not know that it's H5N1, but it could be either Newcastle or H5. All you need to do is to take some bird poop and put it into human volunteers. That's how you start a human pandemic, not with ferrets, not with the mammalian models that we've been using in the lab. So I think we have to keep it real to what is really happening out there.
Thank you for your comment. I'm going to mark that our panel is concluded. I want you to join me and thank all our panelists so much.
And then I guess it's my task to thank the co-moderators, particularly for the very nice presentation. And again, we will now have a short break. We do a 20 minutes break, so I propose we convene here at 4:05, and then we'll have an occasion to study and further continue this discussion in terms of, in the perspective of 4 case studies. Thank you.